Welcome to the Assay Development and Screening Technology Laboratory, where a dedicated group of scientists work to develop methods and strategies in molecular pharmacology with drug discovery applications. The lab has a concentrated effort in cell-based assays involving cell line development, reporter gene assays, and transcriptional and translational evaluation of model cell lines, as well as primary cell culture. Here, the standard assay format involves 1536-well plates. This miniaturized format requires highly efficient and precise liquid handlers to dispense cells and reagents. The multi-drop combi dispenser is the main liquid handler for cells. With its 8-tip peristaltic pump, it is capable of dispensing across a microtiter plate in less than a minute. The EL406 is a 16-tip peristaltic pump dispenser and aspirator. This machine allows for required media or buffer changes in assay protocols and the fixing and staining of cells for high-content imaging in microtiter plate format. The BioRaptor Flying Reagent Dispenser Microfluidic Workstation is a 4-tip piezoelectric dispenser allowing for discrete well dispenses of up to 4 different reagents simultaneously across a microtiter plate in as little as 1 minute. This dispenser is ideal for a host of reagents, enzymes, and buffers. Working in 1536-well format further requires controlled nanoliter volume transfer of compounds or peptides from an arrayed source plate to recipient wells of the assay plate. The principal device for compound transfer is the Wacko-Pin tool, with its aligned head of 1,536 precision-machined notched pins. It accurately and reproducibly transfers nanoliter volumes of DMSO-soluble compounds to the aqueous solutions within the wells of the assay plate. An additional feature of the pin tool is that it will automatically go through a multi-step cleaning protocol within its transfer process, minimizing cross-contamination risk from one transfer to the next. For assays requiring more flexible compound addition, the Mosquito low-volume dispenser is a 16-tip pipetting device capable of transferring 5 to 1,000 nanoliter volumes per well across individual columns of 384-well to 1536-well plates for both aqueous and DMSO solutions. This system is often used in assay optimization and in situations where the compounds may not be soluble in DMSO. The lab employs a number of different detection systems for reading and quantifying various assay outputs, including luminescence, fluorescence, absorbance, laser cytometry, and high-content images. The Vulux is the workhorse of the lab, being the primary reader for luminescence in cell-based reporter assays, luminescence-based cytotoxicity counter screens, and coupled enzymatic assays. The Vulux is a CCD camera-based system where a microtiter plate is imaged with a single exposure and the data is immediately quantified to numeric RLU for each well across the plate. The Tecan Spark is the predominant reader for fluorescence, fluorescence polarization, time-resolved fluorescence, and absorbance assays. The advantage to this machine is that it offers both fluorescent filter and monochromator detection settings, with the capability for top and bottom reads across the plate and supplying on-the-fly data quantification. The Envision is the original fluorescent reader in the lab. This machine employs filters and mirrors for luminescence and fluorescent reads across the plate. Though not as fast a reader, the Envision offers greater sensitivity than the CCD camera-based system, with equivalent on-the-fly analysis. The Acumen laser cytometer offers greater resolution than the previous whole well readers. Similar to flow cytometry, laser cytometry allows for the detection and gating of respective populations of adherent cells, beads, or model organisms including yeast and C. elegans, based on fluorescent intensity and size, and can offer enough resolution to assess protein translocation and distribution of the respective fluor. Distinct parameters can be selected and subpopulations identified and quantified concurrently during the read. The InCell Analyzer 2200 is a rapid and sensitive wide-field cell imaging system capable of reading multiple fluorescent and/or bright field channels simultaneously across a variety of microtiter plate formats and allowing for confocal-like images to be obtained. Often used to confirm or refine fluorescent localization in cells or model organisms, such as C. elegans, high-content imaging is a complementary technique to laser cytometry, offering quantifiable data as clear as what can be observed under a microscope. Supporting assay development and the characterization and assessment of probes, the ADST lab studies the physical basis underlying the biological responses observed in their cellular and biochemical assays, utilizing a range of biophysical techniques and enabling instrumentation. The VIA7 real-time PCR system is a high-throughput qRT-PCR machine capable of reading 96 and 384 well plates for both CyberGreen and TaqMan assays. Beyond validating transcriptional assays, the system is also used for thermal shift analysis to study stabilization of proteins with ligand binding, which have been extensively used in our disease foundation and internal programs. The BioCore S200 is a sensitive SPR-based sensor for the measurement of affinity and kinetic parameters defining the biomolecular interactions between protein targets and ligand probes studied and developed in the ADST lab. The system employs a 4-channel chemically inert gold-coated chip with various immobilization matrices for amine coupling or biotin-avidin-based targets, as well as an ABA injector and microtiter plate compatibility. The system has been employed in various projects including the NSP1 SARS-CoV-2 and ibglycerimide studies. The ADST lab also employs the use of flow cytometry for multiparametric analysis of cellular, bacterial, and C. elegans model systems. The BD Accuray C6 Plus flow cytometer is a compact system able to support a variety of applications including immunology, cell biology, and compound permeability assays. This machine introduces the ability to analyze the expression of cell surface and intracellular molecules, as well as characterize single-cell clones from heterogeneous cell populations. For larger objects, including C. elegans, the Copus Flow Pilot is a flow cytometer capable of measuring and sorting objects in the range of 250 to 750 microns. With its fixed fluidic path, select flow cell, and optics, it can sort and dispense worms or organoids into bulk receptacles or 96 to 384 well microtiter plates, based on size, optical density, and/or GFP fluorescence of individual objects with its 488 nanometer solid-state laser. Another essential element to the ADST program is the capability to express and purify proteins for a variety of projects. Proteins are expressed in E. coli cultures, the cells lysed, and the proteins purified for use in enzyme and ligand binding assays, as well as for antibody production. Proteins are expressed with a His tag sequence purified by single-step affinity chromatography and separated from aggregates on a size exclusion column using the Actipure system. This liquid chromatography system with its low-volume flow path and fraction collector allow for flexible and efficient purification of a variety of proteins, which are further validated with concentration quantification, SDS-PAGE, and Western blot analysis when applicable. Additionally, the ADST laboratory uses automated peptide synthesis to produce cyclic peptides for the ibglycerimide and SARS-CoV-2 NSP1 programs. The Liberty Blue automated solid-phase peptide synthesizer uses microwave energy for both the coupling and deprotection steps to achieve 4-minute cycle times. Peptides can be made sequentially for continuous synthesis. The analytical HPLC in the ADST lab is used to analyze the sample purity and concentration of our cyclic peptides, and when needed for the purification of microgram quantities of precious conjugated ligands. These peptides and ligands can be further aliquoted and concentrated using the SpeedVac vacuum concentrator and define stock samples prepared for later use. Thank you for visiting. For more information about our program goals, operational model, scientific capabilities, and the lab in action, please search ADST at ncats.nih.gov.